Did we finish? Hold on. Do you remember? No. I think we were talking about why Drosophila, correct? Why was Drosophila chosen for the experimentation? So that was our question. That was our discussion. Correct, no? So from there, we had to move on, and I told you Drosophila is a very interesting organism that we use in the genetics, usually in the experimentations of genetics and molecular biology. We use Drosophila, and I also explained you the reasons why. So to revisit this topic, what we are trying to do is, so there were like three people. One is Walter Sutton, Theodore Boveri, and Thomas Morgan Hunt. So what they were trying to do was try to mimic or redo the Mendelian experimentations. But when they did the Mendelian experimentation, they discovered something else apart from what Mendel told us. Mendel's told that through his monohybrid cross and through dihybrid cross, that the alleles exist in pair. The alleles have the property of dominance and they have the property of recessiveness. Correct, right? And also they have the law of segregation, they have law of independent assortment, etc., etc. But if you look at the higher organisms or some of the organisms, they start expressing non-parental characteristics. So when they nobody could explain why these non-parental characteristics even exist and how they are responsible for showing up in the next generation. So the same experiments, when they were tried out, they understood that the genes and the chromosomes are very, very similar. Like in what ways? If you see alleles or genes, they are also in pair, and in chromosomes also, they are the homologous chromosomes are in pair, right? If you see 1, 1, 2, 2, 3, 3, like that, even in homologous chromosomes, we have it in the we have them in pairs. So that's what we understood. So now I also told you that Thomas Morgan Hunt and his colleagues, they started experimentation on chromosomal theory, okay, of inheritance. Again, Morgan was the person, he worked with fruit flies, okay, Drosophila melanogaster. So these two people, that is Thomas Morgan Hunt and Walter Sutton, okay, these guys were responsible for understanding that there is a very, very similar behavior between alleles and chromosomes. Then comes the chromosomal theory of inheritance, correct? No, that is proposed by Walter Sutton and Theodore Boveri. I also mentioned you in the last class that these guys' names are very, very significant, correct, right? So we went through very deeply about what is the laws, why, what are they trying to tell through the, what are they trying to tell us about it, etc. Okay, so today instead of, we wasted a lot of time, guys. That's why I have to just rush a little bit and start with today's topic. So I asked you why Drosophila. So Drosophila was chosen for several reasons, because it is suitable for genetic studies in a very efficient manner. It can be grown in a very simple synthetic medium in a laboratory condition, correct? And they complete their life cycle in a very, very small span of time, that is 2 weeks. Their chromosome number is very, very small, and they can produce a large number of progenies of flies. Male and female can be differentiated very easily, okay, and they show up many of the hereditary variations, okay. So that is why they can be used, okay, to study in genetics. So anyways, so today's topic, sex determination. So we start with sex determination and then we go with linkage. Why? Because when we talk about chromosomal inheritance or chromosomal patterns of inheritance, one best thing that we can visit to understand the power or to understand the contribution of chromosomes or how the chromosomes influence a particular organism. So first we can start with sex determination, and then we can go with understanding what is linkage, what is recombination, okay. So let's see. So when we talk about sex determination, remember Henking was the person. Basically, he tried to trace in 1891. Okay, remember the name Henking. Henking, what he does, he tried to trace that there is a specific nuclear structure, okay, which during the spermatogenesis, okay, is seen in some of the insects. Got it? Some of the sperms had this specific structure, whereas some of them lacked. So whichever had this specific structure, he named them X body. Okay, he gave them the term called as X body. Now you understand. Now, what is it talking about? Imagine, okay, now a particular organism produces, a male produces sperms, right? So when he produces sperms, we know that in male genetics, we have 46 chromosomes. Do you agree? We have 46 chromosomes. Out of 46 chromosomes, guys, 44 are what? Autosomes. And then we have two sex chromosomes. Either it will be XX or it will be XY. But in male, it is XY, correct, right? In male, we have XY. When we go under spermatogenesis, we know that the diploidy condition is converted into what? Haploidy condition. So when they convert into haploidy condition, that means 50% of the sperms, okay, will receive Y chromosome and 50% of the sperms will receive X chromosomes. See, Y chromosome is very small, very small significantly, even in size, in terms of size. So now what happened? He observed that under the microscope that there is a special body that is being seen. So this he called it as X body. Clear, no? This he gave the name called as X body. So later, the same X body is now called as X chromosome. So that is why we call it as chromosomal basis of sex determination. Got it? So first we go for different types of sex determination. Okay. One is XXXO type of sex determination. So what is XXXO type of sex determination? Here we have when we take an example of a grasshopper. Okay. So this is our grasshopper. Now our grasshopper, what he does is there is male and there is female. So the female will have autosomes. Okay. Her autosomes will be what? A and A, and her sex chromosomes will be X and X. Okay. So this is her female. This is a female. Whereas what about the male? Male will have A and A as the autosomes, and then what will be our sex chromosomes? XO. What is O represent? O represents the absence of one chromosome. That means she has, let us say, let's say that the number of chromosomes in diploid state, example, okay, a number of chromosomes in diploid state of a grasshopper is, let's say, 22. Okay, the whole set of chromosome is 22, let's assume. If this is the number of chromosomes that is present in the female, then what would be the number of chromosomes in male? It would be 21. How? For example, if I take female, she will have 20 autosomes and 2 sex chromosomes, XX. Whereas our male will have 20 autosomes and he'll have 1 sex chromosomes, that is XO. So that is how it is. Okay. So XXXO. So this type of sex determination is seen in grasshopper. So the grasshopper contains 12 pairs. Sorry, it's already given here. 12 pairs. I told it 22. I took it as an example. So anyways, it has 12 pairs, that is 24 chromosomes. Got it, no? So male has only 23 chromosomes. Okay, male has 23 chromosomes, whereas female has 24 chromosomes. So let's say that when the spermatogenesis happens, 50% of the sperms, what they will do? 50% of the sperms, they will have X chromosome, correct, no? And 50% will have no sex chromosomes. So whichever the 50%, you can see here, if this is the sperm, 50% of the sperm with X chromosome and 50% of the sperm with no chromosome. So now what will be the female? Female, what she will do? She will produce egg. The egg will have both sex chromosomes. No, 100% of her gametes will have similar type of chromosomal makeup. So that's why she will produce 100% of XX type of eggs. Now this sperm, X sperm goes with X, we will have XX. So XX is what? Female. Again, this X will go, that is X sperm will go and meet the X egg. Then again we'll have XX. So that will give rise to what? That will give rise to another female. Then again, if the O is what? O is another type of sperm. So that means without any sex chromosome. If this sperm goes with this egg, then we'll get XO. Correct, no? And again, this sperm will go with another egg, then we'll have what? XO. You see this? So in the absence of one sex chromosome, it is resulting in the morphology of a male. Okay, the anatomical and morphological structures, everything will be what? Male. Whereas if we have both sex chromosomes, then we have what? A female. Got it, no? So this is about XXXO. And remember the example, it is grasshopper. So the next one is XX, XY type of sex determination. When we talk about XX and XY type of sex determination, it's very, very simple to understand. So why it is very simple to understand? Because we can understand this similar to us. It's in us also, in human beings also, the type of sex determination that takes place is XX and XY type of sex determination. What is another example? Drosophila. In Drosophila also we have XX and XY type of sex determination. Here we have a mother and we have a father. So we know that she's a mother. That's why her genetic makeup, her chromosomal makeup will be what? She's a human being, so she has 46 chromosomes. No, 23 pairs is 46 chromosomes. So she will have what? 44 autosomes. And then what she will have? She will have XX. And what he will have, father will have 44 autosomes. And then he will have XY 6 chromosomes. Correct, no, guys? Now, some of you might be wondering what is an autosome and what is a sex chromosome. Okay, can somebody tell me what is an autosome? Use your chat box. Can some of you guys tell me what is an autosome? Do we have an answer? Okay, I'll wait for your answer. Okay, let's see if somebody gives me an answer. Now, what happens? Mother produces egg. When mother's gametogenesis happens and she will produces egg, all her eggs will be of what? It will be of X type of eggs. Only X eggs will be produced, correct? No? X type of egg will be produced. When a father produces sperms, okay, during his gametogenesis, what he will produce? He will produce two types of sperms. 50% of X type and 50% of the sperms are of what type? It is of Y type. Agree? So, when mother produces gametes, it is of uniform gametes. That is called as female homogamety. Homo means same, gamete means formation of gametes. Okay. Now you see the father. The father is producing two different types of gametes. Correct, no? One is X type of gametes, other one is called as Y type of gametes. So since he's producing two different type of gametes, so we call it as what? Male heterogamety. So they ask you what type of gamete sex determination is done in human beings. In human beings, we see that there is a male heterogamety. That means a male produces two different types of sperms. One is X type of sperms, other one is Y type of sperms. Whereas a female, she produces only one type of egg, which is having all only uniform type of chromosome, that is X type of chromosome. Now we already know if a mother's X egg goes with father's X sperm, what will be born? Female will be born. That means her chromosome should be X and X. Same way for the male, what should be there? For a male, it will be one X and one Y. Then it will be a male. The same goes with Drosophila, guys. So female has X type of chromosome, okay, and the male has XY type of a chromosome. And the Drosophila sex determination is very behaviorally is the same as human beings. Somebody told me the answer. So chromosomes which do not decide the sex is called as sex chromosome. Correct, correct, Sneha. Thank you for the response. Okay, so she is right. So what is the role of autosomes? Now look here, guys. Chromosomes contain bulk amount of genes. Now, you know what? Let's say that at least 5 times small is your Y chromosome. Y chromosome is very, very small compared to that of X chromosomes. Just remember, and just imagine, just the small Y chromosome is responsible for so many masculine characteristics. That is, all the secondary sexual characteristics that is developed in a man, okay, is because of the tiny Y chromosome that is present. Okay, now what is the role of X chromosomes? X chromosome is even more superior, even more significant, I can tell. In what parameters are we talking about? See, your secondary sexual characters in male is hardly 20% of the it's hardly 20%. Your whole makeup of your body, like the basic structure of the body is X chromosome, I can tell. X chromosome, you know what I'm saying? So you see, in the female has two X chromosomes. Okay, and look at what can one X chromosome do? The one X chromosome would give us the inactive breast, that is the chest. Correct, right? So we have that, and then all the small characters like the development of facial hair, the voice cracking, okay, and the other characteristics are all the Y characteristics, okay, Y chromosomes. Now you see the other chromosomal disorders like Klinefelter syndrome, or if you see Down syndrome, now you understand, okay, one chromosome is extra, and see what's happening. It's very uh, drastic change. It's too much change in the biology of a organism. So that's why the superior chromosome is X chromosome always. Whereas even the Y chromosome is very small, you see how much it benefits or how much it contributes for the masculine characteristics that we can understand through chromosomal inheritance. Okay, so let's see another one. Next one is called as ZZ and ZW type of sex determination. So this is a very unique type of sex determination. Why? Because here is completely opposite. So in this oppositeness, what we see is the male produces this, very similar type of gametes, whereas the female is the one who produces two different type of gametes. So let's look at it. So what happens in birds? In birds, especially, you have a pigeon or a hen or whatever it is. What they do is they have the autosomes, and their autosomes uh, along with a diploid pair of our autosomes, the male has, okay, ZZ as their sex chromosomes. So when he produces a sperm, he will produce 100% of one type of sperms that is called as Z type of sperm. Got it? No? He'll produce only Z type of sperm. Whereas let us talk about female. She has, okay, her autosomes are diploid. Along with her autosomes, what do we have? We have sex chromosomes. Sex chromosome is ZW. When she produces gametes, that during gametogenesis, she produces two type of eggs. What is that? One is Z type of egg, other one is called as W type of egg. Got it, no, guys? So now what are the chances? The chances are, if a Z sperm goes with a Z egg, what does it give us? It gives us ZZ baby. The ZZ baby is what? It's a male bird. Again, when the Z sperm goes with a Z egg, what does it give us? It gives again a ZZ baby. Again, it's a male. Whereas when the Z sperm goes with a W egg, it gives us what? It gives us a female bird. Okay, so what do we call the sex determination where you have male gametes which are very uniform, 100% are same, whereas in females we have 50% Z type and 50% as your W type. Correct? No? What do we call it as? So just see here, in human beings, we called it as male heterogamety because male was producing X type of sperms and Y type of sperms. Whereas here, what do we see here? We see that, okay, excuse me. Yeah, we see that the female is producing two different type of gametes. When a female produces two different type of gametes, we call it as female heterogamety. Okay, are you guys understanding this? Yeah? Yes, no? Chalo. Now what happens? There is one more type of inheritance, that is you're the chromosomal number based of inheritance. Correct? No. So in chromosomal number based of inheritance, what do we have a situation here? This is a very unique haplodiplontic sex determination, we call it as, based on the number of chromosomes. We call it as haplodiplontic sex determination. So I'll just write it here. The slide is missing. I'm sorry about it, but we can understand. So the example for haplodiplontic, okay, haplodiplontic, I'm sorry about the handwriting, okay, it's very slippery. Okay, haplodiplontic. In haplodiplontic, what do we have? The example is honeybee, guys. Okay, honeybee. So what do we have here? We have a male honeybee, okay, male bee, and then we have a female bee. Okay. So the male bees in their adulthood, for example, if I check my chromosomes except for the germ cells means the sperms, except for that germ cells, if I check any part of my body, what would be the number of chromosomes? It's obviously diploid number of chromosomes. So what is my diploid state? It is 46 chromosomes, guys. Correct? No? It is 46, irrespective. If I check any part of my cell, it should be having 46 chromosomes except for my germ cells. But it is very, very different in male B. In male B, what happens? It is somatic cells. It's vegetative cells. Like for example, the cells of the eyes, the cells of the body, the cells of the internal organs. Any cell you take and examine, you see that the male B has only haploid number of chromosomes. That is, their entire genetic makeup is what? N, not 2n. They're in haploid state. Whereas the female honeybee, she will have diploid, that is 32 number of chromosomes. How did it happen? Because that's how their entire genetics is made of. So let's see what happens. So we have a male, I will represent it as M, and we have a female. Correct, no? So the male, what is the number? Ploidy. The ploidy is N for the male. Correct, no, guys? And for the ploidy for female is what? 2N. Now what happens? Male have to produce gametes. For the male to produce gametes, can he undergo meiosis? It's not possible. There is an extent you can reduce the number of chromosomes. What is the highest extent you can reduce the number of chromosomes in an organism? It's only N. Oh, what is the meaning of that? For example, if I say that humans have 23 pairs of chromosomes, that means the whole set of chromosomes, okay, in their vegetative state is 46, correct, no? 46. How much can I reduce the chromosome number? Only to its half. That means during gametogenesis, I can only bring my chromosome number to N. There is no way I can have an organism with N by 2 or N by 4. Is it possible, guys? Is it possible? Can somebody tell me why or why not? Okay. Can somebody tell me why it's not possible or why not it's not possible? Can you guys tell me? Why can't I have N by 2 number of chromosomes? Why can't I have N by 4 number of chromosomes? Etc. So you just give me your answer. Okay, not possible. Why? Why is the question? Why is the question? Okay. So now, as you type the answer, I hope you can listen to the further instructions as well. Let's see. So when a male is haploid, he can only undergo one type of division, that is called as what? Mitosis. So when he undergoes mitosis, mitosis is an equational division. So he will produce, okay, an N type of what? Sperms. Correct, no? Next. A female has 2N. She has 32 in her chromosome. Correct, no? She can undergo meiosis. She undergoes meiosis. And she produces what? She produces egg. Got it, no? Got it, right? If the egg and the sperm meet, if the egg and the sperm meet, what does it lead to? It leads to N plus N. If N plus N happens, what does it lead to? It gives us 2N. When it becomes 2N, what kind of an organism do I get? I get a female. Correct, no? If the sperm, okay, if the egg does not meet the sperm, then that will, sorry, if the egg does not meet the sperm, then that will turn into a male. If the egg meets a sperm, that will turn into a female. So whenever there is an addition, sorry, too much I have written. Do I have space? Yeah. If I have an N plus N where the egg and the sperm will come, then it will lead to diploid state. That will give rise to what? A female. If I have an egg which does not meet the sperm, then it will lead to what? N, a haploid male. How does a male, okay, take birth without the fertilization? So what is this process called as? Do you know the process, guys? When a fertilization, when two sets will have same chromosomes, so we can reduce it into half, okay? But if we reduce N, one gamete will not have all types of chromosomes. Absolutely correct. Okay, this is a very smart answer. And again, you have given, which that is Jabroon and Sneha, both of you given your answers as parthenogenesis, and it is absolutely right, guys. And her explanation was very, very logical. That means when I have a homologous pair, if I have 1, 1, 2, 2, 3, 3, 4, 4, all the way till 23, 23, I have another guy, another pair, another couple for me. So one has one. So when I reduce, I can be 1, 2, 3, 4, 5, 6. Correct? No? So how can you reduce 1, 2, 3, 4, 5, 6 into another half? So when I do another half, what she's telling, there is no way you can have a set, complete set you cannot have. So if you don't have a complete set, you know that, right? Even a little bit of changes in the chromosomal structure, number, or position, or shape will lead to a lot of aberrations. So when the aberrations means what? Mutations will come. So when the mutations come, what will happen? Deformities will come. So an organism can never be having the same traits as the parental organism. So that is it. Got it? No? So this is about sex determination. Now, next one is, what about sex-linked inheritance with respect to Drosophila? What did they do? So now what happened? They tried to conduct, it's a very simple topic. They tried to conduct a dihybrid cross that is similar to that of a pea plant. In pea plant, what did we do? We chose two characters. Correct, no? What is that? One is a seed shape and a seed color. And when we did the cross, we got a ratio. What is that ratio? It is called as the phenotypic ratio. Our phenotypic ratio in a dihybrid cross with respect to a pea plant, Mendelian ratio was how much? It was 9:3:3:1. So whenever I do a dihybrid cross, that means what? If I choose two characteristics, okay, and these two characteristics should be what type of characteristics? They should be contrasting characteristics. So if I take these two contrasting characteristics and then I conduct a cross between them, then my phenotypic ratio should be 9:3:3:1. I hope I did not explain you what is a phenotypic ratio right now. Correct? No, and you can, I hope you know this. If you don't know this, please put it in the chat box. We can talk about it again. Got it, right? So that's why they choose varieties which is having two characteristics. What is that? One is a wild variety. So the wild variety of Drosophila has two characters. What is that? One is the eye color, other one is the body color. Okay. So what do we have? We have a red eye and then we have a brown body. Got it, no? And then the same wild variety also has a normal sized wing. Next, we have a mutated Drosophila. So we have to, we can mix the characteristics anyways. It has white eyes, yellow color body, and a short wing, or a miniature wing, we call it as. Now, let's see, okay, what are the crosses and how the crosses are conducted. If you see all these colors, what are those? The color of the eye, the body color, and the wing size, all of these characteristics. For example, the eye color, the body color, as well as the wing size, all of them are present on what chromosome? X chromosome. Very, very important. Very, very important. Okay. Now, let's see. Look here. I have chosen what? I have chosen cross A and I have chosen cross B. In the cross A and cross B, I go with two characteristics each. So what is that? In the first case, what I do is I take eye color and then see here, I have a yellow body with white color eye and then a brown body with a red color eye. Correct? No, one is eye color, other one is a body color. Next one is what I take is I take an eye color and a wing size. So in cross A, okay, I'll write it here. The cross A, what I have eye color, and then what do I have another one? Other one is the body color, correct? No? Body color is my cross A. So these are the two characteristics, guys. So we have two varieties. What are the two varieties? The two varieties are one variety. One variety is what? One is a mutant type, other one is a wild type. So what is our wild type? Our wild type contains what? He contains red eye and brown body. Remember this, red eye and brown body. Whereas when you take the another one, mutated one, it has yellow body and white eye. Now it's very simple. Similarly, like how you take the yellow and round, green and wrinkled. Similarly, we have two contrasting characteristics. Okay. Now what happened? We have the F1 generation, and the F1 generation was self-crossed. Then we get what? We get the F2 generation. The point is, mathematically, when we perform something, it should not deviate from its original numbers. Correct? No? If I take the radius of a circle, then can I change the value of pi when I calculate this, circumference, or when I calculate the diameter? Can I change the value of pi? Is it possible? It's not possible. It's a universal ratio, and you have to keep it as 22/7. It's simple. Correct? No, if you can't take it as 23/7, it's never there in mathematics, because mathematics is truth. So that's what happens here. At some point, when they conducted a dihybrid cross using the same Mendelian two characteristics, contrasting characteristics, they never got 9:3:3:1 ratio. But what happened in the F2 generation, they observed something. What did they observe? Out of the whole population of the F2 kids, they were born, okay, grandchildren, they were born, they saw that 98%, how much? 98.7% were of what type? They are of parental type. They were matching exactly like parents. How much people? 98.7% of them. Whereas, if you see the new varieties were what? They were of 1.23%. Yeah, 1.3% were of new guys. They were not at all looking like their parents, like us. For example, I and you are there, which we don't look like our parents. We look new, maybe taller, maybe black, or maybe white, something like that. So similarly, they were not looking like their parents in the first cross. Now they were like, what happened here? Mendelian ratios are not matching. But what we can see is there are more parental, exactly like parents. There were like many people, and then very small, very minute number of population is having new characteristics, or something is changing here. So next, what they do? They were not satisfied. So next they go for cross B. In the cross B, like that of cross A, what they do is they take eye color, but instead of body color, they take the analysis of wing size. Okay, in a wing size, you know that the wild type has red eye and the normal wing, the wings. Whereas the wild type, what do they have? They have white eyes and they have miniature wings with small wings. When they cross the same way, you have the F1 and then you have the F2. But when they come to the F2 generation, they were very surprised. Why they were surprised? Only, okay, the large, okay, a decent group, I can tell, a very decent group of this new generation were having very new characteristics. How much of them? They were around 37.2. 37.2% of them were new, and new varieties are called as what? Recombinants. Okay, they are called as what? Recombinant. Whereas the percentage significantly reduced in this cross. How much of them? 62.8% of them were exactly matching the parents. Okay, so now it sparked the plug in them. Okay, something is happening here. So what is happening? These guys were very smart. Why they were smart? When they conducted the experiment, okay, when they conducted the experiment, what they knew was, they knew where the genes were located. They knew that it was located on the X chromosomes, and they didn't know the position of the genes. So they didn't know exactly where the position of the genes was. They only knew that these genes are present in what? X chromosome. Now, after this cross, what they understood was, whenever we see that the parental characteristics are continued from one generation to another generation to another, that means that the two characteristics, what are the characteristics? We choose two characters, no? One is the eye color, other one is the body color. That is two characteristics. So the genes responsible for them should be very closely associated. They should be in a very close relationship. Whereas in the other case, whenever we have two different characteristics and they do not have that much deep association or deep friendship or deep bond, that means that is why they are allowing the new characteristics to come in. When you don't have the new very tight bond, new friends will come inside your circle. Correct? No, when you're very close friends, you don't want any other friends. You stick on to each other. So that is called as linkage. So what is happening in linkage? In linkage, we talk about the position of the genes. So here what is happening, if I take in the first case, if I take, this is the X chromosome. Okay, so the position of the genes, the eye color and the body color, they were very, very close or very, very near to each other. That is why separating them was very, very difficult. Okay. Whereas in the second case, if you see, the position of the genes were at quite a distance. So since they are at quite a distance, the separating them or bringing the non-parental characteristics was very, very easy. So what is bringing new characters? What is separating? So usually we know that during meiosis, okay, when the gametogenesis is happening, so we know that there is a phase where a recombinant nodule is formed during the cell division. You have studied in your +1. So during this recombinant module, a process of recombination happens. What is the process of recombination? You see, recombination can be simply defined as bulk transfer of genes. That means like how you have a hard drive to take the whole information out of the CPU, correct, bro? Like you just copy it, a bulk file. Similarly, the bulk transfer of the genes, it doesn't go with one gene, selection of one gene after another gene. It doesn't happen like that. If I take if a recombinant nodule is formed in this location, all the genes that are located in this location will be transferred. As simple as that. You get my point? So like that, a recombination is a bulk transfer of genes. So whenever we have a recombination, now what will happen is, based on the linkage, we will get either it is a recombinant or an parental. Okay, a recombinant variety or a parental variety. So what is the meaning of this? A parental variety is seen when the linkage is very, very strong. When the linkage is strong, when the genes are very close to each other. When the genes are very close to each other, their physical association is very strong. When the physical association is very strong, the linkage is very strong. Got it? So if they are far, then the linkage is weak. When the linkage is weak, recombination is high. Did you understand this concept? That's very, very simple, no? Yeah. So you have to remember few things. What is that? When is linkage strong? When is recombination strong? This one thing. Who conducted the experiment? On what organism they conducted the experiment? Next one is, where is the location of the genes? X chromosomes. You understand me, guys? What is the percentage of recombinants? What is the percentage of no, parental in both the crosses? If you remember these numbers, you're good enough. That's it. Okay. I hope you guys are okay with it. Okay. Chalo. Next one is mutation. Okay. So what is mutation? Mutation can be simply defined as the sudden changes in the genetic makeup of an organism is called as a mutation in simple terms. Okay. Now you see, during the course of evolution, two things are very, very important. Okay. What is important? One thing is the organism should adapt. The organism's genetic makeup should always keep on changing. Okay. Now, what are the two important things that is required for an organism to evolve? Can you tell me what are the two ways in which an organism can evolve? Okay, because can somebody tell me the answer? How can an organism evolve or change? So that is my question. How can an organism change or evolve is my question. Okay. Like while you answer this, I will go. One is, oh yeah, we have an answer already. One is mutation. Okay, so screen is not clearly visible. Bhuvaneshwar guys are telling me this. Is it the same for everybody, guys? Can you see? Yeah, is it the same? Chalo, then what we do is visible. See, people say visible. Yeah, guys, I think you have an internet issue. That's a problem. Okay, I think definitely that's a problem. Okay. See, when we talk about it's internet problem, guys, definitely. Try YouTube. Try coming to YouTube. You can see, check the YouTube if there is a live going on in YouTube as well. So you can just look into it and see if it is visible and it is clear. Okay. So let's talk about mutations. So the two ways in which organisms can evolve is two. One is recombination, other one is mutation. So if you see recombination is Slowly non-parental characteristics keep on adding, and with non-parental characteristics the organism will gain new abilities, correct? No, you can see that maybe you are more intelligent, more smarter, you picked up things very faster, your physiology is quite different from your parents, so we are always advancing as species. So similarly other organisms are also advancing. Why they're advancing? To in order to adapt to different climates, different environments, etc., etc. So recombination can take thousands and thousands and thousands of years in order to create one new character, one new future or something like that. It can be hundreds of years, it can be thousands of years, or it can be hundreds of thousands of years or millions of years for you to see the prominent changes. Got it? No? So this is about recombination. But when you see mutations, mutations are having a bad term because of the cancer, because of the diseases, etc., etc. Of course, mutation is like the sudden change. It's an abrupt change in the genotype of an organism. If the genetics or if the DNA or a particular genes order or the makeup is suddenly changed or shifted or deleted or inserted, anything happens to the genetic makeup, this is called as a mutation. Mutation is always non-directional. There is no direction to its change. It is abrupt, whereas recombination has direction because it's just slow, it is progressive, it knows what to do, whereas mutation don't know what to do. It can happen suddenly, so it is called as non-directional changes in the evolution. But both are significantly important in order to give rise to new characteristics. Okay, so now whenever you have a sudden change in the genotype, what does it lead to? It lead to the change in the phenotype. Okay, got it? So this is called as an organism. This is called as a mutation. Now, how does a mutation occur? A mutation can occur due to the loss. So what is a loss? Whenever a part of a genetic code is deleted, okay, through deletion, or you insert extra codes, that is called as insertion, or you duplicate the same codes, that is called as duplication, okay, of what? Of a segment of DNA which is responsible for giving characters. That is, especially we are talking in the genes, not in any part of the DNA. The genes form of a DNA, whenever it is something is lost, something is added, something is duplicated, then you have a mutation. Got it? No? Or sometimes the whole of a chromosome can be altered. So this is called as chromosomal aberrations. Now, when we talk about gene mutations, okay, so this is can be a single base pair change. That means you have a whole sequence and there is a small one change in one base pair, that is called a single point mutations, we can call it as. Okay, so even the single base pair changes, then either it is inserted or deleted or replaced, again it is called as a point mutation. So here also what do we have? You have a disease or you have a phenotypic differences, etc. Example is what? Sickle cell anemia. Next one is a frame shift mutation. In a frame shift mutation, a whole of a frame, a sequence of a gene is either deleted or inserted. Okay, a sequence of base pairs. So now what will happen? A reading frame will be there. Let's say that you have a bus is coming. Okay. Okay. Now what will happen? You put red here. A red bus is coming. The meaning supposed to be a bus is coming. Now this red doesn't make any sense. Even though for you reading it makes a sense, but in the genetic expression it's a huge change. You understand my point? Now, otherwise you delete the bus. A is coming. There is no bus. Now what will happen? Shift is there. You can see the shift in the reading frame. After, when I say the shift in reading frame, okay, what does it mean? Sorry, what does it mean? It means that after A, B should be there. After B, U should be there. After U, S should be there. After S, I should be there. This is called as reading frame. Because of the deletion, A became beside I. It doesn't make any sense. Even though if it makes sense, the phenotypic expressions will not be the same. You understand my point? So every sequence, what letter comes after what letter? When I say letter, it is about nucleotides. We'll talk about it in molecular biology even more detailedly. So this sequence of letters, you can see here, right? CHANGE. So this sequence of arrangement of letters that is written in our DNA has to be very properly maintained. Whether you put one letter, okay, instead of change, I put S. Okay, you get my point. Only one S is inserted. What might it lead to? It leads to very, very, very abrupt changes. We will see in one of the diseases. Okay, whether I will delete only one word, what is the much difference? That is also point mutation. No, CHA. So CHA will give me one meaning. Then NGE would have given one more meaning. But since the absence of N is there, then it will be what? GEI will come. You get my point? So you understand now. Later you will understand why each letter and each letter sequence is very, very important, which you call it as nucleotide sequences, is very, very important in genetic makeup. So whenever there is changes, whether in a single base pair or a sequence of base pairs by addition, deletion, or duplication, now the reading frame will change. With the reading frame, the genetic sequence will change. With the genetic sequence, the expression of gene will change. With the expression of gene, what does it lead to? It lead to phenotypic variations. With the phenotypic variations, when we observe, we call them mutants. Got it, no people? Okay, did you understand? Yeah. So mutation is a very, very simple and a vast topic as well. As you keep on reading, you get more interesting. But for now, it's enough, I think. Okay. Chalo. Next one. So this is the you know, the slide about mutation. I have shared this a slide with you guys last time. You know, this PPT I had shared anyways. So you just go through this. Next one is a pedigree analysis. So the pedigree analysis is a symbolic representation of how we can trace genetic disorders or any genetic variations or a particular hierarchy or hereditary. We can trace back to several generations. So it gives us a lot of value. Instead of just writing down the names, you can just go with symbolizing from where it started and how it changed and where it has come through. So we can build a whole family tree based on one character. So we can take any one character, whether it is a disease, whether it is a new character, a new feature. We can just pinpoint that one thing for not just this generation, for generation before, we can just trace back and represent it as a tree. And we can also predict what can the future generations be if they marry this kind of person, if they marry that kind of person, etc., etc. We can predict their future very well with respect to how their genetic expressions can happen. That is why pedigree analysis bears a very significant value. What is a pedigree analysis? It's a symbolic representation of hierarchy or a hereditary tree, okay, of genetic traits or hereditary. So this analysis for several generations is called as what? Pedigree analysis. Got it, no people? So this is very, very significant, not only for counseling genetics, people to avoid disease or people to avoid anything. You see, let's say that you are working as a genetist or as a biotechnologist or any molecular biologist. You are tracing one character. It can be a good character also, not only just disease. You can predict, no, okay, this organism has having this character or this trait. What would be the next one if it mates with that one, etc., etc. You can trace it for several generations, and you can also trace its past also. So either way, it's a win-win situation, whether to prevent the disease or predict the disease, or to predict the other characteristics or prevent the appearance of other characteristics. It's a win situation. So that's why pedigree tree speaks a lot of volume with respect to understanding genetics, especially. Okay, guys, I'm sorry. Again, we ran out of time. That's why you have to really come to class on time. So 10 minutes loss is a big loss for us. Okay, I could have finished this topic wholly. So, anyways, I hope you understood today's class. So here it is, guys. Please don't leave the session without filling this. And thank you very much. I'll see you in the next class. And next class, we start with mole basis of inheritance. And I shared you the question paper. You remember? I shared you a question paper to just try and work out some of the questions. Okay. So please let me know if you had any doubts. How did you feel the paper? So was it too much? So that one. And next one, a PPT was shared. And I hope it was helpful. And I hope your AIT exams also went well. So I'll just look into your results and I'll come back tomorrow and I will discuss what we can improve and how we can improve your marks and which areas to focus and everything. Okay. Thank you very much for coming to the session, and hopefully next time you're more punctual and come on time. Okay, guys. Have a nice day. Have a nice session. No, thank you. Thank you. Bye.